Scientific Communication Assessment 1 by Geordie Rowley. GABA stands for gamma-aminobutyric acid, which is a neurotransmitter responsible for sending messages through neurons in the central nervous system. GABA is important as it inhibits or reduces the neuron excitability. This classifies GABA as an inhibitory postsynaptic potential. There are three types of GABA receptors. Ionotropic receptors activate specific ion channels. GABA A and GABA C receptors contain GABA binding sites and a ligand-gated chloride ion channel. The binding of GABA onto GABA A and GABA C receptors triggers the chloride channel to open in the postsynaptic membrane. This causes an influx of negatively charged chloride to enter the cell, where it hyperpolarizes and inhibits the transmission of an action potential. There are many subtypes of GABA A receptors, with each subtype responsible for the binding of different ions and compounds, such as benzodiazepine, ethanol, barbiturates, and anesthetics. GABA A receptors are fast inhibiting, whereas GABA C receptors subtypes are much more selective of what it binds to, and its activation time is much slower than that of GABA A, meaning that its physiological effects are much slower. GABA B receptors are metabotropic. They contain GABA binding sites coupled with separate ion channels by a G protein and as secondary messengers. GABA B receptors are located on both the presynaptic and postsynaptic neurons. Binding of GABA onto GABA B receptors activated the G protein, which then triggers the voltage-gated channels to open. This activation increases the conductance of potassium, which repolarizes the postsynaptic neuron and allows it to make its way back to resting membrane potential. Simultaneously, the GABA B receptors on the presynaptic neuron decrease the amount of calcium entering the cell, which ultimately affects the number of neurotransmitters being released. Both of these actions act to inhibit action potential. Valium is a well-known benzodiazepine recognized for its calming effect. They are sedative drugs that decrease neuron overexcitability present in patients with anxiety or panic disorders. Alcohol is known as a depressant when consumed in high doses, which slows down neurotransmission and causes many physiological effects in the body, such as disorientation, dizziness, memory loss, lack of coordination, and impaired judgment, among many others. Both alcohol and benzodiazepines bind to GABA A receptors on the postsynaptic neuron and increase the frequency of the opening of chloride channels, thus increasing the inhibitory effect on the postsynaptic neuron. Consuming both Valium and alcohol together can be a deadly combination. This is because both drugs cause the depression of the central nervous system, meaning neuron signaling and physiological processes slow down or are inhibited under the influences of either drug. But the combination of both drugs further enhances the suppression of the activity in the brain and intensifies the effect of the sedative components of Valium. This interaction can cause injury due to disorientation or lack of coordination, or the sedative components can cause the user to fall into a coma and potentially cause accidental death. Chloride is critical for the normal functioning of skeletal muscle. Chloride flows through voltage-gated ion channels called CLC1 located on the sarcolemma to help stabilize the electrical charge of muscle cells to maintain normal muscle contraction. During muscle depolarization, potassium leaks out of the cell, and repeated activity causes a decline in the potassium concentration gradient across the sarcolemma. This is because repolarization has not been sufficient enough before the generation of a new action potential. This is where chloride steps in. The large chloride conductance in skeletal muscle allows negatively charged ions into the muscle cells to assist in repolarization before the succession of a new action potential. This maintains muscle membrane potential to avoid muscle tetanus and fatigue. Achannelopathy is a disease that disrupts the function of an ion channel. Myotonia is a disease defined as the hyperexcitability of skeletal muscle due to the electrical instability of the muscle membrane. It is characterized by impaired or delayed relaxation of skeletal muscles. The disease can be caused by a mutation in the CLCN1 gene responsible for chloride transportation, which means chloride is not entering the cell to assist with the repolarization of an action potential. The absence of chloride can decrease the threshold potential, allowing action potentials to be triggered more frequently. Physiological effects of this disease lead to muscle stiffness, weakness, and muscle wasting.